of the protein, and specifically talking about the protein isolation from the yeast. The procedure is also the procedure which I'm going to talk about is also applicable for bacterial cells as well. Okay, and the buffers compositions which you are looking here, like for example the wash buffer, as you can see, it's have got tris, the NaCl, glycerol, EDTA. This might differ again from lab to lab in terms of their concentration, and it all depends, as I said in my previous lectures as well, that it is the way people are getting their results. So this is a stock solution, as you can see, from where you can make a stock solution of NaCl. Now, why someone make a stock solution? Because if someone is using routinely, that's why they make a stock solution, and then a working solution they use it. The analogy which I can give, like in a monthly, you buy your rations or grocery store, certain things you keep it and then slowly use it for 15 days or for a month time. So that's the same purpose that you make a stock solution, higher concentration. But if you see 1 molar, but you're using it for 20 millimolar. So there is a wash buffer. What it is used, we'll see in a moment. Then you have a protein lysis buffer, and I'm assuming here is that the cells has been taken out, whether it is a cheek cells if you're talking about, or you are talking about E cells. You grow those cells into the appropriate mediums, and then you are pelleting down your cells. So that is also been explained. So the difference between a lysis buffer is that you are adding a detergent. That's the only thing which you are doing. You can, some protocols you might find that they have also used in a 0.01% to 0.1% of SDS, or in some places also Tritone X100 has been used. The then one thing is also used, that is the SDS space sample buffer. The SDS space sample buffer is also commercially available. One can buy that, okay, or they can also prepare in a laboratory setting. It's essentially consists of a pH. You can see it is differ from this pH, which is near about the physiological pH, and this is little bit slightly acidic. Then you have a glycerol, you put the SDS, you use a reducing agent. Alternately, one can use DTT also if the 2-beta mercaptophenol is not there, and a bromophenol blue, which is called as a tracking dye. What is its use, we'll see in a moment, but this is to let you know this composition of these things. Now, essentially, there are two types protocols which I will be describing. Very easy to follow. In one protocol which you use it, that you simply lyse the cells by heating using a sample buffer, and the other steps which you can use, that using this protein lysis buffer allow the lysis to work by mechanical disruption, and then you can separate out the proteins, which in part 1 I have already mentioned. So let's say you have a take the yeast cells which is grown, it has to be freshly grown. Preference should be given to the log phase cells. I have already mentioned about the growth curve analysis, that what is log phase, stationary and lag phase there. So you can refer to that. Centrifuge at 3000 rpm, okay, and then you are basically getting your pellet. So something like this, you collect your pellets. So pellets, the cells is been collected, and discard the supernatant. The next step is you are adding, so this is a procedure number 1 which I'm referring to here. You add the sodium hydroxide. Now sodium hydroxide is a alkaline, and the concentration is also used pretty much high. In this basically in that much can say the pH, the cell will lyse. Okay, the cell will lyse at this sodium hydroxide concentration. One can also heat directly, that not an issue, but this is one of the protocol which I am saying. 500 microliter of your sodium hydroxide, keep it at 5 minutes. Then afterwards you will again do a centrifugation step, and in this centrifugation step is something like this, a tabletop. You discard the supernatant. Now you will take the pellet. Now this can then be dissolved, like say the next step which I'm saying, that you can dissolve in the SDS space sample buffer. The SDS space sample buffer, which consists of your DTT, glycerols, your tris, and your bromophenol blue, like this somewhere. You heat the, or you boil the samples, okay, like 95 degrees Celsius in a heating mantle, or you can also use dry heat or also a water bath, and then you can load your samples. Okay, so this is one of the procedure. And to get rid of these things, you can means if you want to do not want to load that, you want to store it for a long time, maybe later on you want to do, then you can also keep it at -20 or -80 degrees Celsius. Now coming to the procedure 2, okay, that is one procedure that essentially you, in the procedure 1, you um, lyse the cell with sodium hydroxide, put in a sample buffer, centrifuge, and just load into the your SDS page. The second procedure, where you are, again the steps is same. You take a 1 to 2 ml of yeast cells. If you are working in some other system, the same procedure, like you can use it to pellet down the cell. So as the same procedure similar to the procedure number 1, then you, in this case, instead of using sodium hydroxide, there is a difference, like you're using a lysis buffer. The lysis buffer again consists of the composition I referred, tris, glycerol, and EDTA, and you have also adding here the NP40 buffer detergent, but as you can use the SDS also. Like people, since you are doing SDS based, that's why people use a NP40 buffer for that. And then you are, now at this time you are also adding a 0.5 millimolar glass beads. Means you, in say, let's say this is your the Appendrof or 1.5 ml tube. You have a pellet. In this pellet you are adding your lysis buffer. Okay, so this is your lysis buffer containing Appendrof. So this is lysis buffer. At the stage step, you are also adding a glass beads, and the size is 0.5 glass beads. It's very small glass beads, okay, which is again commercially available, different sizes. So you just add it here. So this process of lysis step is called as a mechanical lysis. Okay, mechanical lysis. Okay, and for this people either use a vortex machine or they can use a bead beater also. After this lysis step, one has to if you are using a bead beater, you can two way that you can do it at 4 degree, means there are cold rooms. You just keep your samples over there and use the bead beaters for 2 minutes to 4 minutes cycles. If you are using a vortex machine, means you're doing it manually, then you have to probably keep it for a cycle of 45 seconds, and then keep it in ice if you do not have a cold room facility. Cold room is like a separate room where the temperature is maintained at 4 degree. Most of the national laboratories they do have, but if you are in a lab which they don't have the cold room, then you can take the facility of the ice. Just simply you can perform in the lab bench also. After this, you will get rid of the debris by centrifugal force, that is at very high speed for 10 minutes. So this is the centrifugation machine, and this is your bead vortex machine, just showing the sample that how it looks like. And after this step, you will basically dissolve in your same sample buffer, that is your SDS buffer, and then you can load your samples in the gel, and you can run the gel and see what is the protein. So this is a steps, is very simple procedure which can be followed. That is procedure 1 and procedure 2. Procedure 1 is just simply heating and using the denaturant like sodium hydroxide. In the procedure 2, use a lysis buffer, and then you basically can to see the cells. Now say in part 1 we also discussed that there are several other things which is there, that is if you have to do a salting out other preparation. So if you, because this preparation is just to see your whole protein isolation, but if you are preferentially want to follow a procedure, then you have to follow a salting out method, or you have to use an ion exchange, or you have to use affinity chromatography. With this I will stop here. Thank you very much.